everyone, I'm Mukundh Balasubramanian and I'm coming to you from the Slovenian capital city of Ljubljana, and I'm here to talk about my role as an experienced researcher in the Marie Curie Research Training Network interfaces. Within this network, a diverse group of researchers explore eco-hydrological interfaces as critical hotspots for transformation of ecosystem exchange fluxes. My research specifically focuses on developing a rapid and reproducible protocol for concentration and detection of human pathogenic enteric viruses in seawater. This is essential as these water bodies are used for recreational use and for aquaculture. Let me walk you through briefly on how I go about carrying out my objectives for the research within the interfaces network. I am primarily hosted at BioSistemika, a company where a team of molecular biologists and software developers work in tandem to deliver clever and intuitive software for researchers. This starts right from the beginning with the design of each new experiment and the planning of samples that I anticipate and the kind of analysis that I will perform on these samples.
Once the experimental design is finished at BioSistemika, I move on to my secondary host institute, NIB, the National Institute of Biology. Now it's time to enter the lab facilities at NIB, where it's time to suit up. Here I use a specialized chromatographic support, the convective interaction media methacrylate monolithic columns, or SIM columns for short. These SIM columns can be modified to have active site chains with different chemistries that enable efficient concentration of enteric viruses in seawater through ion exchange or hydrophobic interactions. Here we see the SIM concentration in action, where the liquid chromatography unit on the left is pumping seawater samples containing enteric viruses through the SIM column in the middle into the fraction collector on the right. After concentration with SIM chromatography, I move on to detect the viral particles using real-time polymerase chain reaction, also known as qPCR. I manage this part of my research using BioSistemika's Plater, a pipetting assistant that allows me to seamlessly prepare my qPCR reactions. Here we see the qPCR reaction occurring in real time, where a targeted region of the rotavirus genome is being amplified and detected in our SIM concentrated seawater samples. In summary, the powerful combination of SIM chromatography for virus concentration and qPCR for virus detection, synergized with the software-assisted research workflow tools, has enabled me to develop an effective protocol for the monitoring of pathogenic viruses in our precious coastal and seawater ecosystem.